This video is about DNA structure and replication. In other words, it's about the smaller parts out of which DNA is made. It's about how those smaller parts fit together. And it's also about how DNA is replicated or copied. We talked about the CHNOPS elements: carbon and hydrogen, nitrogen and oxygen, phosphorus and sulfur. These are the atoms that living things are made out of. They're not the only atoms found in living things, but they're the most important. The CHNOPS elements combine with each other to form organic molecules. Among the molecules formed by the CHNOPS elements are nitrogenous bases, 5-carbon sugars, and phosphate groups. Well, nitrogenous bases, 5-carbon sugars, and phosphate groups combine to form nucleotides, and nucleotides combine to form nucleic acids. Two types of nucleic acids are DNA and RNA. If the 5-carbon sugar in the nucleotides is deoxyribose, the nucleic acid is deoxyribonucleic acid, or DNA. If you look up here, we have the molecules that combine to form the nucleotides in a nucleic acid. And we see that the 5-carbon sugar in these nucleotides is deoxyribose, and that's why we call this DNA, or deoxyribonucleic acid. DNA is one of the 4 macromolecules. DNA stores genetic information, and genetic information tells the cell how to develop. In the image on the right, we have a DNA molecule. We can see the nitrogenous bases sticking out here. Each of these nitrogenous bases represents one of the nucleotides in this DNA molecule. So there's one, here's another. The nucleotide is the nitrogenous base plus the sugar and phosphate represented in black here. The sugar and phosphate is going to be called a sugar-phosphate backbone. There are 4 types of nucleotides found in DNA, and the only difference between these 4 types of nucleotides is their nitrogenous bases. The nitrogenous bases found in DNA are adenine, thymine, cytosine, and guanine. And adenine, thymine, cytosine, and guanine are usually represented with the letters A, T, C, and G. In a DNA molecule, the 5-carbon sugars and phosphate groups of the nucleotides combine with each other. And the result is called a sugar-phosphate backbone. And so we have that down here represented in black. This is our sugar-phosphate backbone. The nitrogenous bases protrude from the backbone. To protrude means to stick out or extend from. So we have our adenine, our thymine, our cytosine, and our guanine sticking out from the sugar-phosphate backbone. In some images, the sugar-phosphate backbone is just a line. So if we look down here, there's our sugar-phosphate backbone. If we look over here, this is the double helix, so we have 2 sugar-phosphate backbones. And the same thing is true here. So it's represented as just a line there. You can't distinguish the sugar and the phosphate groups. In other images, you can. In other images, the sugar and phosphate groups can be seen. If we look down here, we have a nitrogenous base, and it's a little cut off here, but we have a 5-carbon sugar, and then we have a phosphate group. This is one nucleotide right here. This nucleotide attaches to the one above it, where we have a nitrogenous base and a 5-carbon sugar and a phosphate group. So this is another nucleotide. We know that this 5-carbon sugar is deoxyribose, by the way, because this is DNA. One way we know this is DNA is that we have 2 strands. DNA is double-stranded. But the point here is that the 5-carbon sugar and the phosphate group of one nucleotide, they combine with the 5-carbon sugar and the phosphate of the next group. So this phosphate group here It's going to attach to a 5-carbon sugar. And then there'll be a phosphate group up here attached to this nucleotide. This whole thing will be a nucleotide. And then the phosphate group here will attach to the 5-carbon sugar in the next one. And the point here is this is what's called the sugar phosphate backbone. And that's what's represented as just a line in some of these other images. Just know that in some images you'll be able to distinguish the sugar and the phosphate groups, but not in other images. In a single strand of DNA, the nucleotides can combine in any order. So we have A, T, C, and G right here, but the nucleotides don't have to combine in that order in the single strand. So down here we have A, G, G, and G. Up here we have a different order. Down here we have another order. They could be in any order in a single strand. But DNA isn't found in a single strand. DNA forms a double helix shape, so that's represented over here. It's often described as a twisted ladder, right? So you can kind of see how that would form a ladder and then it's been twisted. And the double helix is composed of 2 strands of DNA. And so we've untwisted the ladder down to this image here. Notice we have, here's a sugar phosphate backbone, and here's another one down here. And here are our nitrogenous bases are in the middle. We saw that the nucleotides may bond in any combination in a single strand. The 5-carbon sugars and the phosphate groups, they're not picky about which nitrogenous base is attached to any of the nucleotides. However, the nitrogenous bases are very specific about which nitrogenous bases they will bond with. So we have T here, we have A here, and we're going to see that this is no accident that these particular nitrogenous bases are combined with each other. Adenine will only bond with thymine. Cytosine will only bond with guanine, and vice versa. Thymine will only bond with adenine. Guanine will only bond with cytosine. We can see this down here. Thymine and adenine and thymine, guanine and cytosine, and so on. This is called base pairing. The nitrogenous bases are pairing up, and they're very specific about which other nitrogenous bases they will bond with, which other nitrogenous bases they will pair with. The 2 strands are said to be complementary, and they complement each other in the sense that each strand has just the right sequence of nucleotides. So up on the top, we have T, A, G, and C. And down on the bottom, we have exactly the right sequence of nucleotides for these 2 strands to combine. This is the only sequence that could combine with the strand on the top, A, T, C, and G. And so in this sense, the 2 strands complement each other. If we know the sequence of bases in one strand of DNA, we can deduce or figure out the bases on the other strand. For instance, if there's an A in one strand, we know that it's bonded to a T in the complementary strand. So we have A here, we know that we have T up here. If we have T here, we know that we have A here. Down here we have cytosine, up here we have guanine. Down here we have guanine, up here we have cytosine. So we've taken a look at the structure of DNA. But for a cell to divide, it must first replicate or copy its DNA. To replicate means to copy. And base pairing is going to be the key to understanding DNA replication. That's why understanding the structure is essential to understanding the process of DNA replication. All living things engage in cellular division. Cellular division is used for reproduction, growth, and repair. There are 3 forms of cellular division: binary fission, mitosis, and meiosis. When a cell divides, it must pass on a copy of its DNA to each of the daughter cells. So before a cell can divide, it must replicate or copy its DNA. And actually, in the case of binary fission, DNA replication can actually happen simultaneously with binary fission. It happens at the same time. But the point is still the same. In order to create identical daughter cells, the cell has to copy its DNA. DNA replication occurs during the S phase of the cell cycle. So here's the cell cycle. We've looked at it before. This is when a cell is born. We have the G1 phase, and then we have the S phase. And we have G2, and then we have mitosis. This is a eukaryotic cell. Well, it's during this phase, the S phase, or synthesis, that the DNA is replicated. So we're about to talk about DNA replication, and just know that it's during this part of the cell cycle that the DNA is replicated. Don't forget that DNA forms chromosomes. So that image on the right, that's of a chromosome, and it's composed or made out of DNA, the image on the left. So when we talk about DNA replication, we're also talking about the process by which a chromosome creates a sister chromatid. Remember, because this image on the right, this is of a chromosome composed of two identical sister chromatids. It's almost like two identical chromosomes. But remember, we call this one chromosome until those two sister chromatids are pulled apart. Just to tie this in further with what we've learned so far, at the beginning of the cell cycle, the chromosomes are unreplicated. That's our image down the bottom left. And during interphase, the chromosomes are uncondensed. That's our image in the middle. And remember, that image on the left, they're condensed there because I wanted to show you that they're unreplicated. But remember, they're also uncondensed still because at the beginning of the cell cycle, the chromosomes are uncondensed. So that image in the middle, that's what they look like for the duration of interphase. And then during M phase, the chromosomes are condensed and replicated. That's that image on the right. And so this process of DNA replication that we're about to talk about, that's how the chromosomes went from the image on the left to the image on the right. That's how we ended up with sister chromatids, because of DNA replication. DNA replication is when a cell copies its DNA. And DNA replication is carried out by enzymes. Remember, enzymes are proteins that catalyze chemical reactions. And the most important enzyme involved in DNA replication is DNA polymerase. DNA replication begins with the formation of a replication fork. A replication fork is where the double strand unzips. So we have a double strand of DNA up here, and then we can see how it's unzipping here, or it has unzipped. This is our replication fork, this whole section here. There are free nucleotides within the cell. So here's one, here's another. By free, I mean they're not combined with each other. Notice these are all nucleotides here too. We have guanine here, we have cytosine here, but these nucleotides aren't combined yet. DNA polymerase moves along the unzipped strands of DNA. So here's our DNA polymerase. We have our arrows leading to the DNA polymerase here. What it does is it attaches the free nucleotides to the strands. In this image, imagine that the replication fork is over here. And here we have our DNA polymerase. We have our free nucleotides. What's happening in this image is this is a new strand of DNA that's been created by the DNA polymerase. So this is our original strand. And over here we have another original strand, and there's another new strand being created. If you can kind of imagine that. But over here we have our original strand with a new strand being created by the DNA polymerase attaching these free nucleotides. Now the nucleotides bond to the DNA strand according to the rules of base pairing. A bonds to T, C bonds to G, and vice versa. So wherever we have an adenine, a thymine, one of the free nucleotides of thymine is attached. The same thing if we have a thymine, an adenine is attached. If we jump over here, we have a guanine, what's going to be attached? Our cytosine. And we're going to have 3 cytosines in a row, and then a guanine will be added here, and then an adenine, then a guanine, then a cytosine. And the DNA polymerase will move along the DNA strand until it's completed. It's created a whole new strand to complement this original strand. So again, as DNA polymerase attaches nucleotides to the DNA strand, a new complementary strand begins to form. And this is our new complementary strand right here. So the DNA molecule is unzipped, and then the enzyme DNA polymerase moves along the unzipped molecules, the unzipped strands of DNA, and it attaches free nucleotides, attaches them to each other and to the original strands, creating a new complementary strand. But how does this result in identical copies of DNA? How does base pairing produce identical copies of DNA? And to illustrate this, we're going to begin with a single DNA molecule. So on our right, we have a single DNA molecule. The first thing we do is we unzip the molecule. And we now have two single strands. And these are our original strands. And we're going to call them original strand 1 and original strand 2. We then add nucleotides according to the rules of base pairing. So we have cytosine on original strand 1, so we added a guanine. We did something similar over here. Notice that original strand 1 begins with cytosine. So here we have our cytosine. And notice that cytosine is the first nucleotide attached to original strand 2. That's going to be the beginning of a pattern that you want to keep your eye on. The second nucleotide in original strand 1 is guanine. Guanine is the second nucleotide attached to original strand 2. The third nucleotide in original strand 1 is adenine. Adenine is the third nucleotide attached to original strand 2. The fourth nucleotide in original strand 1 is thymine. Thymine is the fourth nucleotide attached to original strand 2. The new strand attached to original strand 2, so this strand here, is identical to original strand 1. And the reverse is true as well. The new strand attached to original strand 1, this one, is identical to original strand 2. The result is that each of the new strands is identical to one of the original strands. Moreover, the new DNA molecules, these molecules down here, are identical to each other and the original DNA molecule. The process of DNA replication is used by all living things on Earth and every living thing that has ever lived on Earth. Without DNA replication, there would be no life on Earth. No organisms could reproduce, and multicellular organisms could not grow or repair themselves. Let's take a look at some MCAS questions. A single nucleotide of DNA is composed of which of the following substances? A. Adenine, guanine, and cytosine. B. Hydrogen, a phosphate group, and adenine. C. Ribose sugar, deoxyribose sugar, and thymine. Or D. Deoxyribose sugar, a phosphate group, and a nitrogenous base. And the answer is D. Deoxyribose sugar, a phosphate group, and a nitrogenous base. In a sample of double-stranded DNA, 30% of the nitrogenous bases are thymine. What percentage of the nitrogenous bases in the sample are adenine? Remember, thymine will only bond with adenine. So we know there's at least 30%. But the other piece to this is that adenine will only bond with thymine. In other words, the reverse is also true. It's not as if adenine bonds with thymine and some other nitrogenous bases. Adenine will only bond with thymine. So the answer is B, 30%. What is the genetic material in the chromosomes of an animal cell? A, DNA. Chromosomes are made out of DNA. This is true in all living things. Viruses sometimes have RNA instead of DNA, but viruses aren't alive. So if we're talking about the chromosomes in living things, we're always talking about DNA. This is another easy one. Which molecule typically has a double helix shape? DNA. Which of the following statements describes each new molecule of DNA produced when DNA replicates? Remember, when DNA replicates, we take the double strand of DNA and we pull it apart. And the new DNA molecules will each have one of the original strands, but they'll also have a new complementary strand. So, A, each new molecule is half the length of the original molecule. B, each new molecule has only the coding portions of the original molecule in its sequence. C, each new molecule contains one strand from the original molecule and one newly synthesized strand. Or D, each new molecule retains the A, C, and G bases in the DNA sequence, but replaces the T base with U. And the answer is C. Each new molecule contains one strand from the original molecule and one newly synthesized complementary strand. Which of the following processes relies directly on the complementary base pairing of nucleotides? A, synthesis of ATP. B, replication of DNA. C, formation of peptide bonds. Or D, pairing of chromosomes in meiosis. And the answer is B, replication of DNA. DNA replication unfolds based on the rules of base pairing. The new complementary strands of DNA are created based on the rules of base pairing. This video was about DNA structure and replication. DNA stands for deoxyribonucleic acid. And DNA is made out of nitrogenous bases, deoxyribose sugar, and phosphate groups, which combine to form nucleotides. And the nucleotides are named after their nitrogenous bases. And the nitrogenous bases found in DNA are adenine, thymine, cytosine, and guanine, represented with the letters A, T, C, and G. In DNA, the sugar and phosphate groups in the nucleotides combine with each other, and they form a sugar-phosphate backbone. So that's a single strand. But then the single strands combine with each other to form a double strand. And that's how DNA is usually found. And it forms a double helix, which we saw looks like a twisted ladder. And the strands combine with each other according to the laws of base pairing, A to T and T with A, and C with G and G with C. So those are the only combinations in which the nitrogenous bases of the nucleotides will combine with each other when the strands combine with each other. We call the double strands in DNA complementary strands because they have just the right sequences of nucleotides to be able to combine with each other. When DNA replicates, the two strands of DNA unzip from each other, and we call the site where this happens a replication fork. DNA polymerase is an enzyme that takes free nucleotides from within the cell and it attaches them to the original strands, forming new complementary strands.